Hello, everyone, and welcome to the concept overview on alterations for digestive function. So I'm not going to worry about stomach physiology or intestinal physiology. That's pretty basic. I encourage you to review that in the textbook. We're going to focus on the liver and the pancreas. These are very complex diseases, but if you understand the pathophysiology or the physiology, the clinical physiology, then it makes the pathophysiology actually pretty straightforward. So let's go ahead and get started. All right, let's start with the liver, a little clinical liver physiology. So if you recall from your physiology course and your anatomy course as well too, the liver is basically this blob of cells, and the main cell is a hepatocyte, and most of the functions of the liver occur in the hepatocytes. And so if the hepatocytes are damaged with some kind of liver disease, damaged or injured, we're just going to see alterations in all of these different functions. So as we work through all of the different functions of the hepatocytes, I want you to be thinking about, well, what would happen in the body if this function was impaired in some way? Okay, and that'll just lead you straight into the pathophysiology. All right, first function. One of the important functions of the liver, and it's doing this all of the time, is it takes unconjugated bilirubin, that is a pigment released by dying damaged red blood cells, and converts it to something called conjugated bilirubin. And so let's just briefly review this process. So remember that red blood cells have a 120-day lifespan, and if they start dying off at the end of that lifespan or prematurely in a hemolytic anemia, we'll see that a lot of the destruction of the cells occurs in the spleen, or in some cases the liver, but let's focus on the spleen. We have splenic macrophages who are going to ingest the dead and dying red blood cells in the spleen. And then we start to break down the plasma membrane, and then we're left with the hemoglobin. The hemoglobin gets further degraded or broken down into the globin portion, which is protein, and then the heme portion, which we're going to have to break down a little further. So those macrophages will keep breaking down the heme. We get iron released, and that iron gets released into the bloodstream. It can circulate around. It can be stored as ferritin in the liver or other cells, or it could be used for red blood cell hemoglobin formation. And then we're also left with this pigment that went into forming hemoglobin on unconjugated bilirubin. And the problem with this pigment is it can't really be recycled in its pure form, unconjugated bilirubin, and can't be excreted either. So what happens is when the unconjugated bilirubin circulates in the bloodstream, when it gets to the liver, the hepatocytes take up that pigment into their cytoplasm, and then they convert it into a form called conjugated bilirubin, and that conjugated bilirubin is soluble in the bile that these hepatocytes produce, and so we can get rid of the bilirubin out of the body in the bile. Okay, so now what we're going to do in the pathophysiology is we're going to say, well, what happens to this whole process if the liver is not functioning? If we've seen hepatocyte injury, hepatocyte death, okay, what happens to this whole process? That gives us the pathophysiology, and then leads us to the clinical manifestation, or at least one clinical manifestation of liver disease. Another important function of the liver, specifically the hepatocytes, is to produce plasma proteins. And there's several plasma proteins that the liver produces. One of them is called albumin. Okay, albumin is just a very generic plasma protein. It's fairly small. The liver produces lots of it, and we can measure albumin levels in the blood with a blood test. And the function of albumin is to maintain something called a plasma oncotic pressure. Okay, so let's talk a little bit about that. I know that Dr. Petta has already talked about oncotic pressure a little when she talked about nephrotic syndrome in the kidney chapter, but let's review it here. So this picture here is showing us there's a capillary here, and here are, these are just cells in the tissue, not hepatocytes necessarily. And so this is just, we're just out in some tissue. Okay. So we have water, of course, and we have everything else, glucose and blood cells and all of that. However, we also have albumin, and albumin maintains what we call this oncotic pressure. And the oncotic pressure is a different kind of pressure. Most pressures are pushing pressures, right? You have a certain volume of water, it creates a hydrostatic pressure that pushes against the vessel wall and gives us blood pressure. This is a little different. The proteins, albumin, they attract water. So albumin holds onto water. So it's a little bit more of a pulling pressure, okay? And so as the plasma and the blood circulates through the capillaries, the tendency is the capillaries just want to let water just go seeping out into the tissues, kind of like a soaker hose. Albumin holds onto a lot of that water and only allows a little bit to escape, okay? And so it becomes really important in holding water into the tissues, okay? If the liver's not producing enough albumin, you can kind of deduce what's going to happen next. But that's the pathophysiology, so we'll talk about that in class. Another plasma protein produced by the liver are clotting factors or clotting proteins. There are 13 different clotting proteins. They all have a Roman numeral number. So clotting factor 1, Roman numeral 1, 2, 3, all the way up through 13. You're only seeing a couple of them here. Here you're seeing factor 10 and factor 12. Some clotting factors also have a name, a given name. So factor 12 is sometimes called Hageman factor. Factor 10 doesn't necessarily have its own name. But here's another one here, prothrombin. That's actually factor 1. And actually, I take it back. Prothrombin factor 2 and fibrinogen factor 1. So some of the clotting factors have names and some have numbers. We're not going to have you really worry about all the individual clotting factors. The only time they become relevant are in clotting and bleeding disorders like hemophilia, where somebody's missing one clotting factor. Okay. So now we're just saying the liver produces all of the clotting factors, and in the end, the clotting factors are responsible for making fibrin. Okay, that's the end product of activating all of these clotting proteins when someone is bleeding. Okay, and then fibrin over here forms the meshwork with the platelets, and red blood cells get in there as well too, but mainly platelets get in there and form a stable clot. Okay, so just remember the recipe for a stable blood clot: you need platelets, but you also need fibrin, which requires that you have enough clotting proteins being produced by the liver. Okay. We'll see that the liver is associated with really different types of clotting and bleeding disorders, and we'll kind of get into that in a little bit of detail in our pathophysiology. So we're kind of hitting all the systems here. Here's another system: inflammation. The liver, if you recall, is responsible for producing complement proteins. And complement proteins are almost as important as your white blood cells in terms of fighting infection. And so if you recall from way back at the beginning of our semester, at the beginning of this course in Module 2, you talked about the fact how these complement proteins circulating in the bloodstream, when they get activated during an inflammatory response, they do things like opsonize bacteria, they coat bacteria, which really flags them for phagocytes such as neutrophils and macrophages, so they promote phagocytosis, and then and do several other things as well. And then in the end, these complement proteins, all the C5 through C9 bind together, they will penetrate, say, a bacterial cell membrane and create holes in it, and in the end cause cell lysis. So really, really important for fighting infection. So guess what happens if the liver can't produce enough complement proteins? Another function of these amazing hepatocytes, all these functions are occurring with the hepatocytes, is the conversion of a molecule called ammonia to another molecule called urea. Now, you've already talked about urea. Urea is a molecule that is filtered and then filtered in the kidneys and then excreted in the urine. Well, urea came from ammonia. Ammonia is a molecule, a pretty toxic molecule, that is produced when cells break down protein. So really, when the amino acids get deaminated, ammonia gets released. Now, a lot of ammonia deamination and protein breakdown occurs in the liver. And so the liver has all this ammonia being released. Well, what the liver does really quickly is it buffers that ammonia so that it's not as toxic and creates urea. So take a look. You don't need to memorize the formula, but here's ammonia. What it buffers ammonia with is carbon dioxide, because there's plenty of it in the body. We're always producing carbon dioxide with metabolism. So we will take some ammonia, bind it to carbon dioxide, and we get this new molecule called urea, and then a little bit of leftover water. Urea is not toxic. Urea can be excreted by the kidneys. Okay, so urea is the vehicle by which we get rid of any excess ammonia. Okay, and all of this process here, this all occurs in the liver. And then just some other general things. Recall that the liver is responsible for storing glucose. So any excess glucose in the blood. So here we are in the bloodstream. Here's the liver. So let's imagine we've absorbed some sugars or carbohydrates, and now we've converted all that to glucose. Our blood glucose levels go up. Any excess blood glucose will then be transported into the hepatocytes and then converted to this stored form of glucose called glycogen. So the liver is responsible for glucose metabolism and also glycogen storage. And then, of course, when your blood sugar levels drop, a little after breakfast maybe, then the liver can break down the glycogen and feed some glucose back into your bloodstream so that you don't become hypoglycemic. Okay, so that's important, storage of glycogen. The other thing that the liver does is if somebody needs glucose, maybe they're not eating, maybe they're fasting, or maybe they're starving, okay? Then the liver will take other molecules like fatty acids, amino acids, and it will make new glucose. That's the process of gluconeogenesis. So here we are, we can take lactate, amino acids, glycerol, put them in the hepatocytes, and then produce actual glucose out of that. And we call that gluconeogenesis. So the liver plays a really important role in those two metabolism functions. So now think about all the things that can go wrong if a patient has liver disease. All right, so that's all the physiology. This is a little bit more anatomy, but this is important to understand because it relates to some of the clinical manifestations of advanced liver disease. So we are going to talk about the hepatic portal system, which is basically the veins and the capillaries that are associated with the liver. So here we have a picture, and you have a picture of this in your notes. Things are cut away so that we can see the blood vessels, and these all blue blood vessels are veins. So here's the liver, here's the inferior vena cava, the IVC, headed up towards the heart, stomach, spleen, pancreas, duodenum. And these are all the veins that are draining all of the blood from the digestive capillaries. So we have a couple, this big vein here is the superior mesenteric vein. It's draining blood from most of the small intestine and part of the large intestine. The little one beside it is coming from the last end of the large intestine, the inferior mesenteric vein. And then you can see all the other veins are just draining blood that is going to be deoxygenated, but rich in nutrients because it just came from the digestive system. And before going back to the heart and the lungs, all of these digestive veins go through the liver. Why? Well, mostly because any excess glucose or other molecules can be taken up by the liver and then either stored or converted to glycogen or made into new molecules that the body might need. So very important little bypass going on here. So let's look at the anatomy, the name of the anatomy, and then the blood flow. So here we are. I've made it all white so that you can actually see the arrows, but the arrows are showing the nutrient-rich blood coming from the digestive tract going back towards the liver. And all of the blood ends up being transported in this fairly large vein called the hepatic portal vein. It's about the size of your thumb, heads into the liver with all of this nutrient-rich blood. And then you can see the hepatic portal vein branches into all these little smaller venules and eventually capillaries. And all of the exchange of nutrients and stuff happens in the liver. And then the blood coming out the other side comes out in this vein here and then heads into the inferior vena cava and then can go back to the heart. Okay, so we just have this little bypass through the liver. So, and this picture here just gives you a little bit more detail. It's showing you, and it's more in the text than anything else. It's showing you that the blood is now entering the liver through the hepatic portal vein. There are these capillaries called hepatic sinusoids where the exchange occurs, where glucose goes, maybe into the hepatocytes and gets converted into glycogen. And then the leftover blood, the blood that's circulating through the liver, comes out the other side into the superior vena cava. Okay, and then so it kind of shows you that direction of blood flow. Now, we are going to talk about some of these labs, but I wanted to have just an overall table here for you. We'll go over these in more detail in class, but these are labs that can help us understand liver function. There is no one lab to tell us whether the liver is working or not. So we look at all these different things that the liver either produces or the hepatocytes have inside that might get released if there's hepatocyte injury. So we'll come back and go over this, but I wanted you to just have a little bit of a preview and a reference here, but it's also in your lecture notes. Okay, so then we'll go over to the pancreas, which is pretty close to the liver, but very, very different organs. And we will need to talk a little bit about the anatomy, but mainly then the physiology of the pancreas. And you've already talked about the physiology of the endocrine pancreas, which will play an important role here when we look at pancreatic disease. All right, first off, where's the pancreas located? And this gives us a sense of where the liver is as well, too. So let's look at the liver first. It's tucked up under the rib cage on the right side of the abdomen, upper abdomen, and it's, the diaphragm is tucked up right underneath the whole diaphragm. And then the pancreas is a little hard to see in this picture. It is actually this little yellow spot here because the pancreas is behind the stomach and behind the transverse colon. So it's in what we call a retroperitoneal position, meaning it's at the back of the abdominal cavity. So the stomach is out front, the large intestine is out front, and the pancreas actually lays over the aorta and the vertebra in the back of that upper abdomen. Okay, and it is in the upper abdomen, it's in the upper quadrant, upper half above the umbilicus. And so when people have pain associated with the pancreas, it's usually up high in a line at the level of the pancreas, but then often the pain also radiates towards the back because that's where the pancreas is located. So it can irritate neurons in the back. So if we dig in and look at the anatomy of the pancreas, it's not terribly complex. Let's just focus on the yellow pancreas here. And if you were to look at a pancreas, and we'll look at some autopsy pictures in class, but it just looks like, again, another blob of tissue or cells. And that's pretty much what it is. But they have a little bit more organization than the liver. So in the middle of the pancreas, there is a duct called the pancreatic duct. And coming from or draining into the pancreatic duct are these smaller ducts, and then they terminate or start with these sacs of cells called acini. So let's kind of work at the acini and then go back to the pancreatic duct. So we can't see acini on this picture here, but they would be all around these little ducts on either side of the large pancreatic duct. Here they are blown up. And so this is an acini or acinar sac. And you can see these are secretory cells around the outside. It's like a gland. The acinar cells secrete stuff into the middle of that gland, and then the fluid drains out here and then goes into the main pancreatic duct. Okay. So we secrete stuff in the extremity, stuff gets into the pancreatic duct, and then at the end of the pancreatic duct, you can see it joins up with the duodenum or duodenum, however you want to say it. And there's a little sphincter here, a teeny tiny sphincter called the sphincter of Oddi. And that can close and keep stuff out of the duodenum, or it can open and allow that fluid to flow into the duodenum. So then the question is, well, what are these acini producing? Well, you know this already, right? Oh, actually, this is just maybe this is a little bit more, maybe a little simpler. This is the picture that you have. So let me just reorient you onto this. It keeps jumping around here. Here is my diagram of an acini or acinar sac. And here they are there. Again, mine's just a little simplified. Here's the main pancreatic duct, and there's the sphincter of Oddi. Okay. All right, well, what's in the fluid? That's the big deal here. And again, we're talking more about the exocrine pancreas here, which is associated with the digestive tract. The exocrine pancreas secretes enzymes into the acini. And those enzymes are going to help us digest our food. So these are our classic digestive enzymes. We're going to have protease to digest proteins, amylase to digest carbohydrates, and lipase to digest lipids. Now, here's an interesting fact, and it's in your notes here. Pancreatic lipase is the only lipase that we have. Okay, so if there's problems with the pancreas, you know, we might not be able to digest fat adequately. So keep that in mind. The other thing to keep in mind from a clinical perspective is the pancreatic proteases are such strong enzymes that if they were secreted as active digestive enzymes into the acini, they would auto-digest the acini. So what the body has done, pretty impressive, is they secrete these proteases as proenzymes, also known as inactive enzymes or proteases. And so, yes, we get the proteases being secreted into the digestive fluid here. However, it shouldn't auto-digest the pancreas. And those pancreatic proteases only get activated when they hit the duodenum. Okay. And then there's just one other reminder here that the endocrine pancreas also secretes insulin and glucagon, but you know that already, you know that well, to help us regulate our blood glucose levels. So obviously, if there's really severe problems with the pancreas, we're going to end up losing some endocrine pancreatic function as well, too. All right, then we'll talk more about this in class, but there are clinical measures that we can use to measure pancreatic function, but nothing like the liver per se. So there's just one blood test, and what it does is it gives us an idea of whether or not the pancreas has become injured. And so if the pancreas becomes injured and we start to see those acinar cells release enzymes into the bloodstream instead of into the pancreatic duct, then we start to see a rise in blood or plasma amylase levels and lipase levels. So this is pancreatic amylase and pancreatic lipase. So normally those levels should be very normal because the amylase and lipase should go into the acinar sacs and then into the pancreatic duct and into the duodenum. So they should never hit the bloodstream. But if there's injury to the pancreas or damage, disease, then we might start to see those pancreatic amylase and lipase levels go up. Okay. All right. Well, that is our overview, and that will really help us set the stage for understanding the complexity of liver disease and then pancreatitis, which is the main type of pancreatic disease that we're going to talk about. So we'll see you in class.